Well, thanks a lot, Fuad, and thanks to all of you that are joining from probably many different places in the world. It's actually quite exciting. My talk will be a mix of past work, published work from my postdoc, but also ongoing things that are happening in the lab right now. And I'm very open and looking forward to your feedback because some of it is probably very relevant to this audience. And I'll try to share a bit how we work on marine microbial interactions across scales. Oh, sorry. So, as probably many of you know, every year one of the most outstanding phenomenon occurs in the ocean, whereby organisms that are microscopic to the naked eye start to rapidly proliferate to form those massive blooms that are visible from space. Those organisms are called we particularly focus on the eukaryotic photosynthetic microbes that come in a wide diversity of shapes and sizes. And those organisms are not only rapidly proliferating and beautiful in shape, they also obviously play a major role for our ecosystem services by being responsible for over half of annual primary production, but also serving as the basis of the marine food web. What has become quite clear in the past few decades is that those phytoplankton do not live in isolation and instead establish numerous cell-cell interactions with other members of the planktonic community that can dramatically impact cell fate. Eukaryotic phytoplankton can interact with bacteria, either extracellularly or intracellularly, that can expand their metabolic capacity. Eukaryotic phytoplankton can interact with eukaryotic parasites or viruses that can hijack the host metabolic machinery, leading to cell lysis. And eukaryotic phytoplankton can also establish more exotic cell-cell interactions with other protists that can end up impacting their behavior and displacement into the water column. And even though those interactions happen within a few microns, they obviously do impact phytoplankton cell fate and ultimately the way those phytoplankton accomplish their functions in our ecosystem. And I think that in a way, at least for me, 2024 has been quite a spectacular year for research in eukaryotic phytoplankton symbiosis early in this year, with the publication from the group of John Zer of the UCNA endosymbiosis between bacteria and algae that turned out to be a nitrogen-fixing organelle, which is basically a newly described eukaryotic organelle, which doesn't happen every day. And a few weeks later, the group of Marcel Kuipers from MPI Bremen reported the discovery of a new Rhizobia diatom endosymbiosis that actually accounts for a significant portion of nitrogen fixation in the North Atlantic that hadn't been discovered until now because people looking into nitrogen fixation were mostly looking at the fruiting fraction. And it's, I think, quite exciting to see how by looking into eukaryotic phytoplankton symbiosis, you can actually touch upon concepts that link eukaryotic evolution and cell biology to actually ecosystem function. So in my lab, we focus on the impact of symbiosis on the fate and function of phytoplankton in a changing ocean. And we do so by addressing three main questions. The first one is how to better chart the diversity and distribution of eukaryotic phytoplankton symbiosis in the open ocean, to zoom in on specific case studies to better understand how symbiosis can actually impact phytoplankton development, and more forward-looking how cell-cell interactions will be impacted by climate change. And to address these questions, we combine different scales of experimental setup. First, in the lab, where we use the very controlled and reproducible conditions of model systems to actually study the mechanisms of some of those interactions. We regularly scale up and conduct mesocosm experiments in which we can directly manipulate natural microbial communities, but also very regularly do outgoing work in the open ocean, in particular where we try to increase our ability to study non-cultivable symbiosis, but at the molecular and cellular scale. And to illustrate this, I want to come back a bit on what I've done during my postdoc at the Weizmann Institute of Science in the lab of Asaf Vardi, where using this combined approach, we were actually looking at the impact of host pathogen interactions across different biological scales. And the system we're working on in the Vardi lab involves a eukaryotic host, which is called Geophyrocapsa huxley, which is a haptophyte calcifying coccolithophore, one of those algae that can form those massive blooms that are visible from space. And those blooms are thought, or were thought for a very long time, to be terminated by the infection by a giant double-stranded DNA virus called EHV. And EHV is really the litivirus that was held responsible for the demise of those blooms. However, back when I started in 2017, we were still unable to answer a very simple question, which was how many cells are actually infected during a phytoplankton bloom. And my impression and my thought was that as long as we don't have assays that enable us to rapidly assess how many cells are actively being infected during a phytoplankton bloom, it's going to be very tricky and challenging to actually quantify properly the impact that viruses have on the export and recycling of phytoplankton organic matter in the ocean. So to address this, we started actually in the lab where we developed an assay that enabled us to uncover new aspects into the life cycle of giant viruses using quantitative mRNA visualization. And the idea was really to fluorescently label individual molecules of messenger RNA and to visualize it using imaging flow cytometry. So this is what you see here, where every row is a cell. The first column is actually a bright field image. The second column is the same cell, but excited with a laser that specifically illuminates viral messenger RNA. And the last column is actually illuminating host messenger RNA. And by this way, you can actually very precisely see whether or not your cell is actively undergoing viral infection. And the reason why we say active is because this giant double-stranded DNA virus, once it enters the cell, actually gets its genome transcribed, and it is thus producing viral messenger RNA. So the minute you actually detect this RNA, messenger RNA, it actually means that the cell is undergoing active viral infection. Because this is done using imaging flow cytometry, we can actually be quite quantitative, and this is what you see here, where we actually are looking at the distribution of cells within one single population according to the intensity of the viral messenger RNA, in this case, the major capsid protein. This is 0 hours post-infection, so just when the virus are added into our algae culture, and you can see that all of the cells are below this dotted line, meaning that none of the cells are infected. But very rapidly, when we sample at 4 hours post-infection, you see that there's this subpopulation of cells that start to shine very bright in the fluorescent channel of the viral messenger RNA, meaning that those cells are actively undergoing infection. And what this tells you is really first of all, that there's quite some heterogeneity, that not all the cells in a culture undergo infection at the same speed, but you can also really track the dynamics of infected cells through time. Here, I'm looking only at one gene. We can, of course, look at two genes at the same time. Here, we are looking at one specific time point, which is 24 hours post-infection. Each dot is actually a cell, and they are positioned based on the intensity of the host messenger RNA. In our case, it's a chloroplast-encoded gene called PSBA, which is a good proxy for the metabolic state of the cell. And on the y-axis is actually the viral messenger RNA. And through this actual experience, we were able to show that there was, in fact, during the dynamic of viral infection, a very early phase during which cells were infected, which is actually this top right corner, during which cells are infected, and they don't shut down photosynthetic machinery, but they already start to release newborn virions in the media. So this is an early phase of viral production without cell lysis, which is later on during infection followed by a massive episode of cell lysis, but without viral infection. So we've been able, thanks to this assay, to actually be much more precise on the different stages of the giant virus's life cycle. Because this was done based on imaging flow cytometry, we also used that data to disentangle single cells from aggregates. One of the main phenotypes of algae infection is the fact that upon viral infection, some algae will start to produce more polysaccharides and form those aggregates, but it remains relatively challenging to quantify. And here we were able to, using DAPI staining, to differentiate single cells from doublets from aggregates and to actually track the distribution of single cells versus aggregates through time between 0 and 56 hours post-infection. And very interestingly, we were able to show that during early phases of infection, aggregates are actually enriched in infected cells. And we hypothesized that this could be a kind of defense mechanism from the host, which actually traps cells that are producing newborn virions and removes them from the free living population, preventing the spread of newborn virions in the naive population. I will stop here because this has been published. But in general, once we had this assay, which is actually relatively easy to implement, we of course wanted to go a bit further. And this is a work that has just been submitted, in which we actually tried to harness machine learning to go even further in our ability to decipher the fate of cells during algal infection. What is important is that in the previous experiment, we actually collected a lot of data. We in fact have over 8 time points, 6 different channels, over 100,000 cells. And in terms of imaging feature, we have nearly 400 features per cell. What we were trying to do was to build trajectories of where the cells are going through time. And thanks to machine learning approaches, we were actually able to better decipher the causes of cell death. Because what is important to understand is that during viral infection, actually not all cells die from viral infection. Some cells are going to die from metabolites or I would say signaling molecules that are released by infected cells that are going to induce programmed cell death into neighboring cells. So there is a fraction of the population that is going to die, not due to direct viral infection, but due to signals that have been emitted by infected cells that will kill those cells. But it's, again, very hard to decipher the origin of those 2 different types of cell death. And by using those approaches of machine learning, we were actually able to trace the origin of cell death and mainly to quantify what was the origin of cell death, whether or not they died from viral infection versus from programmed cell death. So differentiating cell death with or without viral infection. So that was quite exciting, but all of this was done in the lab using the very controlled system of 1 host and 1 virus. And of course, we wanted to scale up. And we decided to conduct a mesocosm experiment. Our mesocosm experiment took place in Norway in May 2018. And the goal was really to try and better link the dynamics of algae bloom and their viral infection and the impact that it could have on ecosystem dynamics. So we basically started by filling 7 bags of 11,000 liters. We draw water from the fjord, which is known to contain cells of our coccolithophore of interest. We basically spiked in some nitrate and phosphate, and we followed it for 23 days. And this was, of course, a huge group effort in which we had many different types of questions that were asked on that same system. So very luckily, here you actually see an average of the population dynamics, looking specifically here at eukaryotes. You have days in the x-axis from 0 to 23, and you are looking at the succession of species here, microeukaryotes. And here in bold is actually Emiliana Huxley, which is the former name of Gephyrochapsa huxley, but it's the same thing today. And you can really see how those cells actually bloomed throughout the middle of the mesocosm before demising. And we actually have this data kind of demultiplexed for the 7 different bags. Because we had this assay of the single molecule fish that enables to label viral messenger RNA in single cells, we decided to adapt it to be able to apply it to natural populations to see if we can track viral infection dynamics in this mixed population. And so we kind of modified a bit the previous assay to add a 3rd fluorescent probe, which specifically tags the 28S ribosomal RNA of Emiliana Huxley, so that we could actually gate our host of interest out of this mixed population and then actually quantify the percentage of infected cells through time. And this is exactly what you're looking at here. From day 11 to day 23, we're looking at the proportion of Emiliana Huxley cells that are actively undergoing infection through time. You can see that there's quite a drastic jump from day 17 to day 18. But interestingly, even though we do see a lot of viruses in our mesocosm bags, we never go beyond 25% of infected cells. So they say that viruses are causing the bloom demise might be a bit of a stretch. They are definitely associated to bloom demise, but they are for sure not the only mechanism of cell death. In a bloom, a crash, and we know it by now, we have pathogenic bacteria, eukaryotic parasites, of course, sinking, programmed cell death, and viruses is actually just one of the many mortality agents that can cause cell death. But first, this was exciting because it's the first time we actually got those numbers. And then what this actually enabled us to do was to compare the percentage of infected cells in our 7 different mesocosms. And quite interestingly, we actually had a big gradient. And because we had a gradient with bags that had no infection and bags that had high infection, we could actually start to quantify the impact that viral infection could have on biogeochemical processes. And one of the processes that we decided to focus on is, for example, the accumulation of TEP. TEP stands for transparent exopolymer particles. These are polysaccharides produced by phytoplankton that are responsible for the aggregation and production of marine snow, and they can play a major role in carbon cycling. Here you are looking at actual measurements of TEP concentration through time across the 7 different bags. And because we had the information of how many cells were actively undergoing infection, we were actually able to better explain the dynamics of TEP that we had in the bags. And one of the conclusions was that indeed algae that are infected by viruses tend to produce 10 to 4 times more TEP than non-infected cells in those bags, thereby explaining why in the bags where we had more infection, we also had more TEP per Emiliana Huxley cell. So that was very interesting. And maybe for some of you that are more in the marine microbiome community, what was very interesting for us also, and counter to some of our expectation, is the increase of eukaryotic Throstochitris during the demise phase of the bloom. When we started analyzing the data, we worked under this classic, I would say, succession in our brain that when we have a bloom followed by a bloom demise, all this particulate and dissolved organic matter is going to fuel the growth of heterotrophic bacteria in the system. And when we started measuring bacteria, we saw that they were increasing, but not as high as what we expected. And then we started looking at eukaryotes, in particular microeukaryotes and osmotrophic eukaryotes, and realized that Throstochitris, which are also osmotrophic eukaryotes that can feed on dissolved organic matter, were significantly increasing to the point that they even challenge this idea that bacteria can recycle the dissolved organic matter, or at least they are probably not the only ones that are playing that role in the ecosystem. We went a bit further with this idea of quantifying the link between single cell and ecosystem processes, and also applied single cell transcriptomics to those cells that were infected by viruses. In particular, we would take sample from those mesocosms, go to a flow cytometry facility, conduct single cell sorting, followed by single-cell transcriptomics. And thanks to single-cell transcriptomics, you can actually recover both the host and the viral transcriptome within individual cells, which really enables us to, for each individual cell, say very precisely at what stage of viral infection it is. We actually have pretty powerful markers, in particular viral genes, that are genes that are typically expressed at the beginning, at the middle, or at the end of viral infection. So you can really say for which individual cell where it is in that I would say viral life cycle or viral cycle. So this is what we did, and this is exactly what you're seeing here. We are looking only at infected cells, individual infected cells on day 19. And we are looking at whether or not they are an early versus late stage of infection. Day 19 is pretty much at the end of the bloom crash or in the middle of the crash. And you can see that most of the cells that are infected are already late in infection, which actually makes quite a lot of sense. We are at the late stage of the bloom. Infected cells are more or less at the late stage of infection. What is interesting is if we go back in time, including in day 15, which is really at the beginning of the bloom, and we realize that the numbers don't change. Still, most of the infected cells are actually already in late stage of infection. And what this suggests for us was that there's in fact a turnover of viral infection, that every day there is a steady state of cells that are being infected, that lies, but that proportion doesn't actually change through time. And for us, what this really indicated is that we really need to start decoupling, in a sense, the fate of the cell with the fate of the population. And it's very clear by looking at this data when you think that at the early stage of the bloom, you already have most of the infected cells that are late stage of the bloom. It's just that the density, the concentration of cells that are infected is relatively low, but you really have to decouple the fate of single cells with the fate of populations. We also went for a more untargeted approach. Here, this approach was really based on single cell sorting, looking specifically for cells that were calcified, which are Emiliana Huxley. Actually, not only the calcified cells, but we were focusing specifically on Emiliana Huxley. But back then in 2018, my kind of bet or dream was that we could use those single cell approaches to actually detect new pairs of host and viruses, because thanks to metatranscriptomics, we can see that there's a huge amount of giant viruses in the ocean, that many of them are being expressed. But because of bulk sequencing, we completely lose the information of who are the actual hosts. And to address this, we decided to try 10x genomics, specifically focusing on the size fraction of 5 to 40 micron. And thanks to that, it just took us 6 years, but we were able to recover new pairs of host and giant viruses, and this has been recently published in Nature Microbial. So this was more of the mesocosm side of the study, and in 2021, I was fortunate to lead one of the expeditions along the Patagonian shelf break in Argentina, where we specifically located coccolithophore blooms using satellite imaging and actually conducted Lagrangian sampling to follow the bloom and demise of a specific natural coccolithophore bloom to pretty much apply the same approaches. And here, stay tuned, this is currently ongoing analysis. So I hope that with this example of the host and the virus, I managed to show you how we can really go from the lab to the mesocosm, to the natural environment, and how it was so critical for us to have this very simple assay of tracking active viral infection within single cells to end up linking it to much bigger processes, in the case, for example, of TEP accumulation through time. So fast forward in 2022, so 2 years ago, I moved to EMBL, where I started my own group. And I will switch gears a bit because we no longer work on coccolithophores, but decided to target the Chaetoceros diatom. That I believe is a really ecologically relevant and scalable model system to think about phytoplankton symbiosis across scales. So Chaetoceros belongs to the big family of diatoms, which are silica cell-walled eukaryotic phytoplankton that actually are responsible themselves for 25% of annual primary production on a yearly basis. Chaetoceros is defined by this relatively simple rectangle and those spines that we call setae made out of silica, and this actually gives them their name for horns. Chaetoceros is a relatively large genus of around 400 species. Some of those strains are single-celled, some of them can form chains. Those chains can have different morphologies. And I find it as an ecologically relevant system, given that Chaetoceros is one of those diatoms that can make those blooms visible from space. But what makes it particularly interesting for me is the fact that Chaetoceros is one of the rare eukaryotic phytoplankton that has been reported to establish cell-cell interactions with pretty much every organism or every class of organism in the planktonic community. They have known epibiotic protists, known nitrogen-fixing ectosymbiotic bacteria, eukaryotic parasites, single-stranded RNA and DNA viruses, but also a well-characterized microbiome. On top of that, it's quite compatible for lab field research. Most of the species grow quite well in the lab. We have some CRISPR available for some of them, and omics resources are quickly ramping up, especially with now a whole project around genome sequencing of representative species of the Chaetoceros tree. Just a brief insight on its life cycle. Chaetoceros belongs to the centric family. These are homothallic species. So during its cycle, undergoes mitotic divisions and as a consequence of the silica cell wall, the size distribution of the population decreases through time to the point that Chaetosaurus cells will hit what we call a sexual size threshold, which induces the production of male and female gametes. Those male and female gametes will conjugate to form what we call a zygote, followed by an oxospore. And the oxospore actually restitutes the initial cell size of the population, and then it undergoes another cycle of size population shrinkage, etc. But the fact that it's a population and a species that can produce in the same strain its own male and female gametes also has an advantage that we can maintain them in the lab for quite a long time. In the natural environment, it has quite an interesting life cycle also. During bloom formation or at the end of the blooms, in general, nutrient depletion actually induces the formation of spores. Those spores are extremely resistant. They have a very thick silica cell wall. They sink to the bottom of the ocean where they can actually remain viable for thousands of years. And upon mixing events, they are actually brought back up into the surface ocean where they start to bloom, engaging in a new cycle. In a new life cycle. What is quite interesting is that all those life cycles actually are also impacted by microbial interactions. And for example, there's some amazing work showing how viruses can actually induce the formation of resting spores in Chaetosaurus cells. So for today, I will mostly focus on one aspect of the research, which is really focusing on our efforts to increase ability to study uncultivable interactions directly in the natural environment, and focusing on one specific case study where we look at the Chaetosaurus interaction and try to understand how it can impact the development of the phytoplankton. And for this, I see in diatoms. As you know, most of planktonic diatoms are actually non-motile. Of course, we do have motile diatoms, the benthic ones, but most of the open ocean diatoms and planktonic ones are non-motile, at least in the largest proportion of their lifetime. This comes with a cost. The cost is that non-motile diatom, you sink out of the photic zone. You cannot really explore nutrient spaces. There is no flight mode when facing threats such as predation, and actually limited dispersal for seed population. But diatoms have found a way to actually become motile, and some of it is actually through cell-cell interactions, in particular with unicellular ciliated protists. This is what you see here. This is a viscous diatom that has elements that are and the hypothesis that they make in this paper is that the ciliates and the flow that's generated by the ciliates actually decreases the diffusion boundary layer around the diatom, increasing another type of very cosmopolitan interaction between the Fragilopsis diatom and tintinid ciliates that is actually quite widespread in the ocean. And the Fragilopsis are actually dividing to form this barrel, which nearly serves as an armor. It's quite, it seems quite bulky to move around with, but definitely there is some signaling going on between the diatom and the ciliates. And we see this repeatedly in our sample. Hypothesis is that it will.
I think we have an issue with the connection, Flora.
Can you hear me?
Yes, it went for a bit. Yes.
Should I cut my video?
If you think that works better, maybe.
I think we should try that, Flora.
I can cut.
Yeah.
Yes.
Okay, I cut the video. Do I need to go back one slide or it's okay?
I think it was an interesting slide. You can go back to it. Yeah, sure. Thank you.
So sorry, which one? Just to make sure.
Previous one.
This
Yes.
one or?
Yes.
Okay. Cool. So very briefly, this lack of motility can come with a cost, which is that we, as a diatom, will sink out of the photic zone, the nutrient mode when you face predation, but also limited dispersal in terms of population genomics. But diatoms have found ways to become motile, mostly through cell-cell interactions. And here what you see is a paper from EMBL from 2021. We are looking at a Cocinodiscus diatom, which has several Vorticella ciliates that are attached to it. And the hypothesis that they make in the paper is that the flow generated by the cilia actually reduces the diffusion boundary layer around the diatom, increasing its uptake of nutrients. In work that I conducted during my PhD, we report the prevalence and abundance of another type of interaction involving the Fragileopsis diatom, which forms this kind of barrel through cell division. And in this barrel, there are different types of tintinid ciliates that actually really drag this barrel around them. And though it doesn't seem optimal in terms of displacement, it could definitely bring advantage when it comes to protection against grazing. And here it's very clear that there is some form of cell-cell communication between the diatoms and the ciliate. However, the challenge of uncultivability. Diatom-ciliate interactions are actually quite prevalent. They are distributed globally. They involve several species of diatoms, several species of ciliates, but they are really poorly understood. If diatoms can be relatively easy cultivated, ciliates are actually very challenging to maintain. And as a consequence, we are lacking quite some knowledge on how specific this can be, what is the mechanism. We've never really recorded the full life cycle of such an interaction, and we don't really know how it compensates and whether it compensates for non-motility in diatoms. And so our thought back then was, well, if you cannot bring the ocean to the lab because you cannot cultivate those ciliates, why not try to bring the lab into the ocean? And for this, I want to briefly introduce the Trek expedition, which took place in the past 18 months and that recently ended, and I was lucky to be part of it. Trek stands for Traversing European Coastline. It's an 18-month expedition that was led by EMBL in collaboration with the Tara Ocean Schooner. And the goal was really to explore the interface between terrestrial and marine environments all along European coasts. And one of the aspects of this Trek expedition was what we call which was a unique opportunity to bring. Can you hear me? Okay.
More or less.
More. Oh, wow, that's annoying. Okay. So
it's okay. I mean, it's okay. Now we can hear you.
Okay. So the Advanced Mobile Laboratory is this semi-trailer, which actually is around 22 tons, which has two panels in the center that open to give rise to a 40 square meter laboratory. And it's equipped with instruments such as high-pressure freezer for electron microscopy sample prep, a confocal microscope, and an imaging cell sorter. And my lab has been particularly focusing on the moment where the Advanced Mobile Lab is side by side with the Tara vessel, so much so that we can actually conduct a very and organisms of interest while getting the whole description. We decide to focus on key players. Because one of the challenges of working with samples like Tara is obviously that these are extremely complex. And we've been harnessing the presence of this imaging cell sorter to actually move from samples that are very hard to work with because complex and full of sediments to samples that nearly look like a pure culture. And throughout the whole Trek expedition, we've been focusing on Chaetoceros that we found pretty much all along the European coastline. And the purification and concentration of that specific genus really enabled us to conduct advanced assays that are usually impossible on Tara. And this has proven itself to be extremely useful in one specific case study, which is work from my student, Thomas Bevis, where by coming back from one of our sampling expedition, we were lucky to stumble upon a symbiosis between diatom and ciliates. It's actually an interaction between the Chaetoceros diatom and pyrethric ciliates that we think belong to the Cossonia family, which has never been described. And we rapidly processed those samples through the imaging cell sorter, and we were actually able to disentangle free living from host-associated cells. So thanks to that, we were actually able to build a gate. And once you can build a gate, you can start sorting for many different types of downstream assays. But first and foremost, we were able, thanks to the high throughput imaging, to actually quantify the prevalence of that interaction, just to see if it was ecologically relevant. By looking at the fraction of diatoms that are found in symbiosis, we were able to show that throughout 3 consecutive days, nearly 15% of the Chaetoceros were associated with the ciliate. And by looking at the ciliate side, we were actually able to show that 100% of the ciliates were attached to Chaetoceros. So there was no free-living ciliate in the samples that we actually collected. And on top of that, all of the ciliates that were attached were always attached to Chaetoceros and to no other phytoplankton. So quite abundant, but also quite specific. Thanks to the ability to sort in quite high throughput, Thomas was actually able to test different culturing conditions and to keep it in the lab for long enough to record its life cycle. What you see here is actually a ciliate just after division from the mother cell that has settled on a new Chaetoceros. And within this 1 hour time lapse, it's actually building its lorica. And by recording the life cycle, we were able to show that the ciliate still divided, but also that the Chaetoceros cells still divide, demonstrating that this definitely does not stop or halt the life cycle of both partners, and that it's therefore not parasitic. We were also able to record a lot of tracks, in particular to try and quantify the behavior of this kind of holobiont to see how it may impact displacement of diatoms. And because we were also able to high pressure freeze concentrated samples directly on spot, we also now are currently analyzing 3D focused ion beam scanning electron microscopy, in particular to focus on the attachment point. You've seen how fast the attachment point can be created, and now we're actually zooming in on this specific area to understand if the stalk building is actually mediated by cilia, whether or not these are actually motile cilia that are repurposed to build this very strong stalk, and this is really ongoing work. Maybe of interest to this community is also how we've been trying to go a bit further in looking at motility. In particular, by analyzing those tracks, we were able to look at different patterns of motility throughout the different phenotypes that we have. And one of the ideas is to see if this newly acquired behavior can actually prevent or reduce encounter rate with predators for the diatoms. So that's really what we're trying to test right now. We unfortunately don't have the predators in the system to test it. But the idea would really be to see if we can plug in this data into models of predator encounter and see if by being attached to ciliates, the diatoms can reduce their encounter rate. Last but not least, and this is really preliminary work, as you can see, we're also trying to conduct PIV analysis. This is super preliminary and super raw, but thanks to the many specimens that we were able to sort, we also conducted some analysis to try and see if potentially this would also reduce the diffusion boundary layer around the diatoms, kind of coherent with the hypothesis that Eva Canso formulated in her paper. If this is also a call to this community that I know is way more expert than me on those questions of microscale biophysics, but if you have tools or ideas of tools and analysis that we could do, it's very welcome. We are still starting there, so there's a lot of room to take smart decisions for the future. So all of this was actually based on samples that were collected literally in a couple of days, thanks to the presence of the Advanced Mobile Lab. But where we were very excited is that thanks to the data that was collected by Tara in parallel, we were able to interrogate data from all over the European coastline to see if this interaction could be found in other places. And this is what we did by using ecotaxa data, where we actually saw that even though we found it in Tallinn, we could actually recover that exact interaction in many different places in the Baltic Sea and up to Roscoff. This is actually only data from 2023, so we are currently waiting for the data from 2024. But this was, first of all, quite encouraging because it showed that it's not just one isolated event that we found, but that it's actually quite widespread. And then what was even better is that we continued getting samples from our local collaborator. And the following year in 2024, pretty much at the same time of the year in June, we recovered that interaction again. So it's not only widespread, it's also a reoccurring event, which also gives us confidence in the fact that it's ecologically relevant. Just some of the few hypotheses, because I get those questions quite often, hypotheses that are more or less easy to test. We do think that maybe the ciliates would benefit from bacteria that are attached to diatoms for feeding. There's also the idea that ciliates, by being anchored to the diatom, actually reduce the drag that is generated by the cilia beating, and that this association with the spines of Chaetoceros could also reduce predation from copepods. On the diatom side, it could be that the cilia are actually decreasing the diffusion boundary layer around the diatom, and this hopefully could be tested by PAV. And if we are lucky, maybe even by SIMS analysis, if we manage to recover the interaction this year, could be that the ciliate also maintain diatoms in the photic zone, so having an impact on their displacement. Could be that ciliates are also enabling diatoms to escape or to reduce predator encounter rates. Could also be that it increases the dispersal of Chaetoceros, which is particularly useful for homothallic strains. And again, some of those hypotheses we are currently testing. Some of them will depend on our ability to recover new specimens in the coming years. But if you have also some additional hypotheses that you can think of, I'm very, very open to hear about them. So in a nutshell, we try to combine different approaches to study the impact of symbiosis on the fate and function of phytoplankton by first increasing our ability to study uncultivable interactions, but at the molecular and cellular scale, but also to establish an ecologically relevant model system and its associated toolbox directly in the lab. We use Chaetoceros genus that I think is both ecologically important, but also extremely relevant for cell-cell interaction studies because it's known to harbor all this diversity of interactions, and hopefully we will learn much more in the coming years. And I just want to finish by advertising for a symposium a bit related to that topic that we are organizing next year, which is called Wild Frontiers of Model Organisms, and will be held at EMBL Heidelberg. And the idea is really to try and see how we can better combine traditional model system-based research with ecological questions. What do we call model organisms, and how far are we from bringing more and new and more relevant systems into the lab? And you're very welcome to submit or to reach out if you have any questions regarding that system and that symposium. That's it. Thank you very much. Sorry for the internet connection. I hope you could hear me well most of the time, and I'm ready to take your questions. Thank you.
Thank you very much, Flora, for this exciting talk. We have 10 minutes for questions. We can take more also, but yeah. Marco.
Yeah, so thanks a lot for the talk, which was fantastic. Yeah, too bad for the little glitch there, but
I'm sorry.
it just was a really nice talk. So I sort of got a little bit lost in the bit on the difference during when you were talking about the viral evolution of the viral infection in the population, the difference between the population level and the individual level, because it looked like your data would be compatible with a standard, if you want, progression of a cell becoming infected, and then late infection, and then it dies. And essentially, the whole population sort of shrinks down because fewer and fewer cells are present, even though, in a sense, the ratio of the early versus late infection keep being the same. I mean, just the proportion doesn't tell you how many cells are there. So I was wondering if you can sort of clarify this point a bit.
I think maybe we had a wrong assumption when we started looking at the data, but I think we didn't expect that proportion of early versus late stage of infected cells to be constant through time. We kind of expected that at the beginning of the bloom, we would have most of the cells in early stage of infection, and then that at the end of the bloom, we would have most of the cells in late stage of infection. The fact that
wouldn't that depend
very
also
early on,
wouldn't that depend also on how many viruses you start with? I mean, if you
yes,
overwhelm the
absolutely.
system.
So yes, and so well, in this case, we didn't add viruses, right? The viruses that infected this mesocosm experiment are either viruses that were already there, maybe hiding somewhere in some copypot poo or coming from the air, because these are different sources of those viruses. What we did observe is that, and this is what I was saying with the fact that I don't think that viruses induce algal demise, but I do think that initial viral load controls or at least is correlated to the maximum carrying capacity that you can have of an algal bloom. So it doesn't control the timing of when it demises, because what we see in our 7 different bags, even the ones where we don't have viruses, is that all the blooms crash at the same time. So it could be that this crash is actually determined by, I don't know, nutrient limitation, or it's not very clear at this level. We would need to do transcriptomics. But what does seem to happen is that initial, or at least first detected viral concentration or maximum viral load during the exponential growth phases of bloom definitely controls the maximum level of algae that we can have. But yes, you're right. Maybe we had the wrong assumption when we started analyzing the data. One thing that we didn't do with that data, mostly lack of time, is to actually plug those numbers into models. I think that would be the natural following step to see if what you say is right. I would have liked to actually test this now that we have that proportion of early versus late stage. It I guess could be so straightforward to make a model and look how it influences cell density and viral concentration, and this we didn't do, and I think that would kind of address that for sure.
Thank you.
Yeah.
Any other questions? Okay, I'm just a question from me. So I was very curious with those interactions. So do you have any idea how they established these first interactions, like with the viruses, or also in the symbiotic one, how they get too close to each other? Because there is no motility, or in most cases there is no motility. How do you?
So in the diatom ciliate one, it's actually relatively easy because the ciliates are motile, right? When loricated ciliate divide, you actually have a zooid that is being expelled from this lorica. The zooid doesn't have a lorica, so it's basically a ball that is full of cilia, and we have a few tracks looking at the dispersal of those cilia. They seem to swim quite erratically, but at some point, and I'm pretty sure it's mediated by some kind of chemical gradient that is released by diatoms, they do get around specific cells. And here in the settlement operation that we kind of managed to capture, you can really see that this zooid, the cilia daughter cell, is really searching kind of its perfect landing spot around the diatom. It's really moving around it. It's definitely not random. And you can really see that it starts to move around the diatom to the point that it starts screening a specific area, and then that area gets smaller and smaller and smaller, and then it stops. And then within 1 hour, it is rebuilding its lorica. We have this kind of hypothesis that it's also triggered by the presence of bacteria, because of course the cell wall of diatoms is covered by bacteria that could be a food source for cilia. But in the case of the cilia, it's relatively easy because they are motile. So this is how they can actually direct themselves towards phytoplankton. In the case of viruses and viral transmission, it's a good question. I mean, viruses are not motile, right? So I guess it's really based on encounter rate. There was this crazy hypothesis that I think this is true in maybe, I don't remember which other system, but in metazoans where infected cells release virions that are embedded in some kind of polysaccharide matrix. And because the viruses are embedded in a sticky material, they actually adhere better to naive cells, and this is how they infect them. And so that could also be a potential option in our system where we already know that infected cells produce more TEP. So you could imagine that on the one hand, producing TEP potentially can make aggregates that remove newborn virions from the naive and free-living population, but it could also be a good way to actually tag and kind of throw your TEP-coated viruses to naive cells and increase the probability that they enter. But viruses don't swim, so for sure they need to find another mode of dispersal. Yeah.
Thank you. In the one that were the other symbiosis relation, like with the Chaetoceros, and we have this multi-form, like bigger aggregate. So this does not impact their sinking rate, or they counterbalance by motility or something?
So for sure, if anything, it will. So we don't have a gravity machine, right? So we cannot see whether or not it compensates sinking. But given the trajectories that we record, these are XY trajectory. I wouldn't be surprised that it would be possible in Z axis. So yeah, my impression would be that this is the type of interaction that can maintain Chaetoceros closer to the surface than sinking. And one of the ways we can actually address this is to compare high throughput imaging data from surface versus deeper layers, and see what's the probability of bumping into that symbiosis in the surface versus in the deep, which I think we have the data for it.
Thank you. Any other questions for the remaining people? I still have one question though.
Go ahead.
Another.
It's only us.
I saw that. So you are using the flow cytometry. So you have actual quantification of cells, and you have using RNA expression. Do you have also like some genes that you are targeting or something? I'm asking to see if you try to correlate the population or an estimate of the population based on the gene expressions or the mRNA expression, something like that.
You're talking about the diatom ciliate system or the viruses?
Any of them, in which in any system where you had a flow cytometry data and RNA expression or gene expression data.
If we are trying to see if we can use bulk metagenomics to quantify cells.
Yes.
No, we haven't tried. No. I know some people are trying to do that. I think it's possible now, but no, we haven't done that.
Yeah, well, it's just given us like here on using environmental DNA, it's a hard time to effectively estimate the population size or something based on that.
Yeah, I know that now, for example, at least for photosynthetic organisms, you can use a PSBO, which is a single copy gene that is present in photosynthetic organisms that has been used also as a marker for quantifying cells. Yeah.
One more final, like large global question. So in many of these associations, there were some nitrogen involved, or nitrogen, at least the bacteria, there were some organelles, or have you looked at like nitrogen at some points and their role?
Yeah. So for
Yeah.
So in the case of the diatom ciliate, it's quite interesting because it happened in the Baltic Sea during the spring and summer, which is extremely stratified and very nutrient poor and nitrate poor. So we do have some single cell transcriptomic data that is going to come. And one of the ideas would be to look at a differential expression of nitrate transporters in free living versus host associated. And if we manage to catch this interaction again in 2025, the idea would be to do SIMS. And if just to test and to compare nitrate phosphate and at least uptake and carbon uptake between host associated and free living. I'm pretty sure nitrate plays a role, maybe not major role, but I'm also pretty sure that none of those interactions happen just for one specific reason, right? It's a bit like the demise phase of the bloom. It's not just viruses that cause it. So I wouldn't be surprised that indeed nitrogen plays a role. Yeah.
Thank you. Any other questions, or we're at the hour now? Okay, so thank you very much, Flora, for this exciting and this is a
I'm
great...
sorry again for the internet connection.
It's okay. Thank you very much. I'll see you everyone in 2025. I will stop